Hi everyone, my name is Keelan and I'm a PhD candidate at the University of Rochester. I am going to be telling you about a recent paper from our group in the Mechanical Engineering department that looks at the question, what drives brain waste clearance? So your brain cells produce a lot of waste. They are very active cells, and just like your kidneys filter waste from your blood to keep you healthy, your brain also has to remove the waste it produces every day. If that waste builds up, it can cause serious problems like Alzheimer's disease. So we're still learning exactly how this process works in humans, but we do know that the brain has tiny fluid channels that carry cerebrospinal fluid, which is a clear liquid similar to water. You can see a video of this on the left, where there are these highways that deliver fluid from big fluid reservoirs into your brain, around your brain, and then deeper into it over time. We think these flows can help wash away waste, and in part that's because we've seen changes to this system in diseases like Alzheimer's. But there's a debate. Does waste leave the brain mainly because of organized fluid flow, or just because of random molecular motion, known as diffusion? An example of diffusion is shown below. This is the process where molecules move around randomly until they're evenly spread out and mixed. So some researchers think that diffusion alone may be able to clear waste from the brain. So we would like to simulate this situation to see how much fluid motion actually matters. But to simulate flow through these fast highways, we need to know precisely where they are. It turns out these fluid highways are around the arteries and veins in the brain. So here's a video of a mouse brain. The arteries are shown in red, and the fluid spaces around them are shown in blue. Since we have a lot of data about mice, we've decided to use detailed maps of the mouse arteries and veins to model how fluid might move both around blood vessels and then through the surrounding brain tissue, where flows are too slow to measure directly in experiments. We ran 3 types of simulations, one with no flow through brain tissue, one with slow flow, and one with fast flow. And in all 3 scenarios, we did allow for fluid motion through these highways surrounding arteries and veins. We tracked how quickly a waste molecule is cleared out over time, and we found that with no flow, the waste barely moves. That doesn't match what happens in a healthy brain. With very fast flow, the clearance was unrealistically quick, but with slow flow through brain tissue, our results closely matched what previous experiments in mice have shown. We then zoomed out to look at what happens across the whole brain. We know from previous experiments that pressure differences driving overall brain fluid motion should be very small. So we wanted to look at how fluid flows into brain tissue might affect these pressure gradients using a very simplified model. We found that when we allowed flow to slowly leak into brain tissue in our model, the pressures looked realistic. They were quite small. But if we force the fluid to stay only in these highways around arteries and not to enter brain tissue, the pressure shot up by about 100 times, far too high to make sense biologically. So we think that because these big variations in pressures in the brain are not observed in mice, this scenario where there is no fluid flow into brain tissue could not represent a live animal. So what does this all mean? Our results suggest that the fast flow around arteries does eventually trickle into brain tissue and form a much slower but still organized flow, and that this slow movement may be a key to clearing waste. Understanding this better, especially in human brains, could help us learn how diseases like Alzheimer's develop and how we might treat them. Thank you very much for listening. If you'd like to read more about our simulations and our results, you can scan this QR code to see the full paper. And I also wanted to thank our funding sources. This research was supported by the US Army, the US National Centers for Complementary and Integrative Health, and the Brain Initiative of the US National Institutes of Health. Thank you again.